Hello, this is Christine McGowan. It's March 28th, 2023. Okay, so I'm going to be talking about the endomembrane system and how different hormones and proteins are moved throughout the cell after they're created. So here is the image that I created drawing the endomembrane system, right? So you have this cell, and inside you have the main components of the endomembrane system are going to be the rough ER, the smooth ER, the Golgi apparatus, and the transport vesicles. So if you're talking about a steroid hormone like progesterone or testosterone, it would begin in the endoplasmic reticulum with the production of cholesterol. There it would be moved to the smooth ER, so that the cholesterol would be made by the ribosomes. Then it would move to the smooth ER, where it would be converted, and this newly synthesized hormone molecule would travel from the smooth ER via vesicle to the cis face of the Golgi apparatus. Here it would be modified and packaged and then put into another transport vesicle, where it would then be released to the outer membrane, and the vesicle would fuse with the cell membrane. So I drew a picture of this through exocytosis, where the vesicle is then moving towards the cell membrane to release the contents of the transport vesicle out to the outside of the cell. And for steroid hormone, the ultimate fate would be to kind of bind on specific target cells to create some kind of physiological response. The steroid hormones can either be metabolized by the body or excreted by the body as well. If you're talking about something like insulin, it would begin in the rough ER as well, where the ribosomes would create it. Eventually it would travel through the smooth ER and then end up in the Golgi. The Golgi would then package it and transport it as well, and it would be released as well through the cell membrane. Insulin can either be used, metabolized by the body, or degraded, or it could also be bind to specific receptor cells as well and be used as the body needs it. The synthesis of cytoskeletal proteins is also going to begin with the ribosomes on the rough ER. And just a reminder that the ribosomes are created in the nucleolus of the nucleus. So then the ribosomes attach to the rough ER. That's what gives it that rough texture. So then the cytoskeletal proteins will be made by the ribosomes in the rough ER. They're then moved over to the lumen of the rough ER, where these proteins are going to be folded. Remember, with proteins, the most important thing about their function is their structure. So with the cytoskeletal proteins, the folding and modifying of these proteins is going to be what ultimately gives them the purpose of the protein. So then after they're created in the ribosome, they're going to then be sent to the Golgi apparatus via these transport vesicles as well. The transport vesicles containing the protein will fuse with the cis space. Again, they'll be modified and sorted and put in specific packaging, and then they will leave via the trans face of the Golgi apparatus. From there, it will move within the cell, different than the other ones, because the cytoskeletal proteins, their purpose is to either perform different cellular processes in the cell. They can either aid in the function of day-to-day stuff and just create more structure for the cell as just the base cytoskeleton, or they may be more involved and they may make more of these when the cell's getting ready to divide, because cytoskeletal proteins become such a vital part of the growing and dividing of the cell. So from there, those cytoskeletal proteins will leave, and then they'll go ultimately to wherever they need to go within the cell. If there are too many of them, they can be degraded by proteasomes, but for the most part, they would go to the cytoskeleton.